to start talking about Mendel, his experiments, and heredity in general. We start out by looking at what was believed a long time ago, this idea of the blending theory of inheritance, where parental traits were basically lost or blended, joined together in the offspring, and that intermediates always formed. We, of course, now know that this isn't the case because everyone would look alike. What Mendel did was he looked at characters that displayed discontinuous variation. These are traits that are inherited in very distinct classes, like purple or white. There's no intermediates for these pea plant flowers. He found experimentally that these traits were not blended. When he combined purple with white, he didn't see lavender. This idea of discontinuous variation, these very distinct classes, is not very common in humans. We do see it in the widow's peak. We see much more commonly continuous characteristics, which we also call polygenic, which are controlled by multiple genes, and we see that in things like skin color and height. Now, Mendel is considered to be the father of genetics and did his research in the 1800s, in the mid-1800s, and he selected this simple biological system. He used very methodical processes, and he used quantitative analyses, and he used very large sample sizes. These are all very important to why he stated these fundamental principles of heredity. Now, we now know that genes are carried on chromosomes, and remember, at this time, they didn't know that. We know that genes are the fundamental units of heredity, and that we can replicate them, express them, and that they can be mutated. What we find here is that Mendel's ideas formed the basis of this classical, otherwise known as Mendelian genetics. And he used the garden pea. Pea hybrids could be produced. A lot of varieties were available. They're small, so they're easy to grow. They mature quickly. Pea flowers on a single plant have both male and female parts, and they can naturally self-fertilize, otherwise known as they are true breeding. And they can also be cross-fertilized. And these are all important points. Some of the terminology I am not going to spend a lot of time with because really these are vocabulary words. When we talk about cross-fertilizing, we're talking about male and female gametes from different plants. Self-fertilizing, male and female gametes from the same plant. True breeding, offspring created from self-fertilization. Hybridization, mating two true breeding plants. A character is a heritable expressed attribute or an observable feature similar to a trait. So we often will use trait and character interchangeably. And the locus is the position of the gene. Now we have three basic stages in his experiment. First, to produce the true breeding strains for each of the 7 traits he decided to study. Then he cross-fertilized those true breeding strains with alternate forms, purple and white flowers, for instance. And then he allowed those hybrid offspring to self-fertilize for multiple generations. He then counted the number of offspring showing each form of the trait, so how many were purple and how many were white. When we look at monohybrids, mono means one trait, for instance, what we've been talking about, flower color, and hybrid means 2 different organisms. So a monohybrid cross is studying 2 variants of a single character. Now, Mendel produced true breeding plants for 7 different characters, and these are the characters that we have listed here. The variants are listed right here. Again, not necessary for me to read them to you. You're welcome to pause and write them down. Pause. Pause. Okay, so now we're talking about the parental generation, and this is how we designate that P sub 0. That means parental generation. These are the true breeding plants. This is where the offspring have just one allele for the trait, meaning they are homozygous for that trait. This is what is used in those first generation crosses that we talked about. Now the F1 generation is when we take those 2 true breeding plants, remember our purple and white flowers, and we cross them. That first generation is the one that he counted to see how many white and how many violet flowers he had. And this is the first filial generation. What he found was that all of the F1 plants looked like just one of the parents. So the visible trait in the F1 trait is the trait that is dominant. The other trait must be recessive, therefore hidden in that generation. He did not see any blending, no intermediates. Now the F2 generation is where he took the F1 generation and self-fertilized them. And what we see is the recessive trait comes back. So now we see both purple and white plants. And then he counted the proportions of those traits. And what he found was that there was a 3-to-1 phenotypic ratio. Phenotypic means, what do I see? And then after many, many experiments with this, he found that it was always the same. Now, it's not going to be exactly, because this is nature, but it always was really close. So we have 3 dominant to 1 recessive. Now, the parent generation is homozygous for the trait being studied. We know that because he created true breeding lines. Then he cross-fertilized these true breeders by hand to get this F1 generation. The F1 generation all looked the same. Then he allowed these plants to self-fertilize. And when they self-fertilized, that F2 generation showed both traits back again at a specific ratio, 3-to-1. Now these are the results. I'm going to encourage you to pause at the end. I'm going to go through some of this fairly quickly, but I would encourage you to pause so you can look at it in a little bit more detail. What we see for our F1 offspring is that it's 100%, and that ends up being whatever was dominant. And then the F2 generation, literally, these are his results, counted purple and white, tall, short, round, wrinkled. So he counted each one of them. And then we do the math and we get these ratios. And we see that they're not perfect 3-to-1 ratios. A little above 3, a little below 3, but all really close. And that is how all of these worked. And we can see that these, again, are the traits that he was looking at. And these are the details. You can go ahead and review that on your own. We have to deal with a little bit more terminology. We've got the phenotype, and these, again, are the things that we can see. The genotype is the underlying genetic makeup. The homozygous means that at any given gene, the alleles are identical for that gene, so they're exactly the same. Heterozygous at that gene means that you've got 2 different genes. So homozygous would be like each chromosome having brown eyes and brown eyes, where heterozygous would be like having brown eyes and blue eyes. When we start talking about genes for flower color, we see that there are 2 variants for our pea plants. And remember, we're looking at the terminology now, so you do need to make sure you're comfortable with that as you move on. But we see that they are heterozygous, meaning that they have the 2 different alleles for flower color. So that means that the gametes produced through meiosis are going to have either the dominant or the recessive. Remember, in meiosis, these are going to get split up. Now, if it's homozygous, they have exactly the same. So the gametes produced here are going to have the exact same gamete because that's all they have to give. So our F2 phenotypic generation, we see 3/4 of the plants with the dominant form, 1/4 with the recessive. That's our 3-to-1 ratio, dominant to recessive. So we self-fertilized each of those F2 plants. Why did we do that? Think about that. And really what we're doing is we want to see what the genotype is of each of these purple plants. We know the genotype of the white-flowered plant because it's recessive. He didn't know that yet, though. So he went ahead and did it for all of these. And then we get this F2 genotypic ratio. We get 1/4 of them are true breeding, meaning that we are getting all purple offspring. Half of the plants were non-true breeding. So you see some purple plants and then the recessive shows up also. And then the last one, the recessive trait, was also a true breeder and got all white flowered plants. And here we see a 1 to 2 to 1 ratio. And these are very specific ratios for very specific circumstances. And that is when we start out with 2 true breeding plants and we go through this process. So here's our 3 to 1 ratio. Remember, the phenotype is a physical appearance. And we see we've got 3 purple to 1 white. But these 4 plants have a 1 to 2 to 1 genetic or genotypic ratio. And that's what we found by this self-fertilizing. So we see that the genotype, remember, is the set of alleles, could be purple and white. Each flower is diploid, so they have 2 alleles. Some are going to have 2 purple alleles. Some are going to have 2 white alleles. But some are going to have 1 of each. And this is where we get our 1 to 2 to 1 ratio. 1 to 2 to 1. Our conclusions are, or his conclusions were, that his plants did not show blending, that the traits remained discrete, and that for each pair found in the parent generation, one of those traits was dominant and the other one was recessive. We see pairs of alternative traits were seen among the progeny of any specific cross, and that those traits were expressed in that F2 generation in the ratio of 3 to 1. Pause. 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 So this is the results. These are his conclusion, that the parents transmit discrete factors that we now call genes, that each individual receives 1 copy of a gene from each parent. Remember meiosis? And then when the sperm and the egg join together, they're each bringing a copy of that gene or a copy of that chromosome to that zygote. And not all copies of a gene are exactly the same. There are different versions: blue eyes, brown eyes, green eyes. And when we talk about things that are homozygous, it means they have the same allele. If they're heterozygous, it means they have different alleles. Remember, homozygous, 2 brown eyes; heterozygous, a brown eye and a blue eye. We also know that the alleles remain discrete, no blending. We also want to remember that just because the allele is there doesn't mean that we're going to see it. It's not necessarily going to be expressed, because the recessive allele can be hidden by the dominant allele, and we see that in the heterozygotes. Now, if there's a dominant allele, it is absolutely expressed. The phenotype, remember, is the physical appearance, and the genotype, the set of alleles. Pause. So as you're looking at this, is it homozygous dominant, homozygous recessive, or heterozygous? So this is one of the points that we need to make sure that we also understand. This is how we abbreviate things. If it's a capital letter, that means it's dominant. If it's a lowercase letter, that means it's recessive. So this is homozygous because it's the same, and it's capital, so it's dominant. So try the rest. Okay, so let's come back, and I'm going to look at this one this one, this one, this one, because they're all the same. They have one dominant, they have one recessive. That means they're heterozygous. We have two capitals, two capitals. That means they're homozygous because it's exactly the same, and it is capital, so it's dominant. And then we have two lowercase, two lowercase. Those guys also homozygous because it's the same allele, but it's the lowercase letter, so it's showing the recessive. Now, the principle of segregation is based on the idea that the two alleles for a gene will separate during gamete formation. Remember, that's meiosis. They're getting one allele from each parent, and we had that randomness, so it's random during fertilization which things line up. And this is literally the physical basis for allele segregation. Now, what's interesting about this, and what I think is so amazing, is that Mendel had no knowledge of any of this. We did not know about chromosomes. We did not know about meiosis in the mid-1800s. So these are things that we have the benefit of that he did not. The first law is the law of segregation, and this is where two copies of a gene separate as a diploid individual makes the haploid gametes. Again, this is meiosis, but this is his first law.